problematic. Problematic pathologist. This is what I was called when I was insisting on checking the quality of tissue image analysis data at the beginning of my digital pathology career when I was supervising tissue image analysis algorithm development. Now, validation and quality check and really making sure that the data you're producing with your image analysis solution is an integral part of any tissue image analysis project, but it wasn't always that way. This video is a talk I gave at the Digital Pathology and AI Global Engage conference in New York at the Aiforia Workshop. Aiforia is a tissue image analysis cloud-based platform, which I used and highly recommend. And the work we did together resulted in a publication that you can use as a blueprint for how to check the quality of your tissue image analysis solutions. So let's dive into it.
Who's doing image analysis in general? Most of you. Okay, good. I don't do that much image analysis anymore, but I used to do a lot. My name is Alexandra Żuraw. I am a blogger, YouTuber, podcaster, and toxicological pathologist. I am the host of the Digital Pathology podcast and run the website Digital Pathology Place. And I know some of you are receiving my emails. So I am super excited to see my digital pathology trailblazers here. So it was 2019. I had my first kid was not even a year old, and I went to this conference, Pathology Visions. Who's been to Pathology Visions? Okay, couple of people. So this is like the Digital Pathology Association conference where all the digital pathology vendors come, and Aiforia goes there as well. And I had a babysitter, and she was with my son, and I was there. I wanted to present a poster. That was the first year of my podcast. So I thought I'm going to go to this conference, and there was an Aiforia presentation by Thomas Westerling Bowie, and they gave food as well, which was fantastic. That was a breakfast presentation, and he said those words: "We will not do the validation for you. We can enable you to do everything else, but of course we're not going to do the validation for you." I'm like, this is my person. He knows that validation is important for image analysis. And now it's kind of no-brainer, but 2019 and let's say from 16 to 19, that was still the time at conferences when you would go and present your results from your training data. So there was not this concept that you actually need a test set and a validation set and that you cannot just overtrain and show that it's a fantastic model. And why this was important for me, that he said that we will not do the validation for you, you actually have to do it yourself. And we have a feature there, and Gillian showed the feature. So he says that, and I'm like, oh wow, super cool. Where did this come from? I started my digital pathology career after my residency. I'm a veterinary pathologist like Gillian as well. And after my residency, this was my first job in a digital pathology startup in an image analysis company. And it was a fantastic feeling because I came from a residency program, and everybody who did this, any type of advanced training, master's, PhD, whatever, when you're the students, you're like at the bottom of the totem pole, let's put it that way. And you only have people to look up to, and maybe you teach students, right? And then you join a company and you're the pathologist or you're the expert and your expertise is there. So it's a fantastic feeling. But then another thing is happening. People start questioning your expertise. People who like have no background of your background start like asking you, are you sure these like plaques are real? Maybe it's not real. Are you sure this is a macrophage? And things like that. For a pathologist or for any expert, it's like no-brainer, right? But people start questioning, and I joined a team where there was no pathologist. So my image analysis scientists, and the setup was similar to what Gillian said in Aiforia, that they had scientists, they have pathologists, and now you have annotators and other team members, like executive members. So my image analysis scientists had access to a pathologist before I came 1 day a week for a 1-hour consultation. And basically the concept was that pathologists, it was still the times before deep learning. It was when pathologists would go in and annotate where the analysis was supposed to take place, and the image analysis scientists would design an algorithm and run it on those images. But there was not really any feedback loop if this analysis was done in the correct place. Now you have less need for that, although I do not agree totally with that, but there is less need because the subject matter expert already is giving the annotations. They are already giving their brain onto the slide, and this is what you train them all on. Before, it was sitting with the scientists and trying to figure out an algorithm that is the most generalizable, right? So that led to a couple of adventures that I had in this company that where different teams were not on the same page regarding the quality of the solution, of the image analysis solution. And it led to a conflict, and the group that I had the conflict with were not really the image analysis scientists. We had a really nice working relationship. I learned a lot from them about how to develop image analysis. They learned a lot from me about pathology, and then we were pathology of those images, about the stains. We were later doing similar stains, so they like built a body of knowledge. So there was a good relationship, but there was another group. And are there any biostatisticians here? Biostatistics, bioinformatics, data analytics? Yeah, so that was the group. That was the group. That was a dangerous group. Why do I say it was a dangerous group? Because they take the data and they like work this data so that the data tells a story. And if the data is good, if the data is robust, if the data is accurate, yeah, that's what they're supposed to do, right? But there was a workflow in place where there was not really any supervision. What kind of data would go into the data mining, into the data, working the data, let's call it working the data. And I joined and we started working on quality. And it turned out that this data should not have been mined and no signatures should be generated from this data, signatures. So we were working to support immune oncology portfolio for various pharmaceutical companies. And the holy grail is to find a signature, as a tissue-based signature that can separate the responding patients from non-responding patients when it comes to some therapy that the companies are working on. So there was some fantastic data and fantastic separation presented to various stakeholders that when the tissue was examined, when the image analysis results were examined, the signal didn't really come from a relevant place. So like in life, garbage in, garbage out, and then you work the garbage hard enough and you come up with fantastic conclusions. And it's not specific to that particular situation. I just happen to be in that situation. But like if anybody did PhD, master's, whatever work in the lab where you are the owner of your project, like at some point, it's on you that the quality is there, right? I was doing some PCR for my PhD, and at some point, I'm not that like, I couldn't follow through all the way, but at some point when I was pipetting for hours, I was like, I could add so many different things there and have so many beautiful bands. Like I could make this experiment be whatever I wanted, right? And anybody can do that if we don't pay attention to the quality of what we're doing, and not on purpose, also by accident. So that was my adventure with the data. It ended up with the quality of data. It ended up with conflict, a pretty serious conflict where I became somebody not easy to work with. And at some point, I said no to a releasing of some data, and the data made it out of the group anyway. And that led to a little bit of a debacle. But then it led to putting in place SOPs and putting in place workflow, putting in place a separate quality control group in the whole company that assured that no garbage would go out. And it was especially important because the data was used, or was the algorithms were being prepared to be used in clinical trials. So it wasn't preclinical anymore. I mean, not that preclinical is less important. I'm a toxicological pathologist. It's super important. But it wasn't anymore the research and development space. It was supposed to be used on patients, on single patient samples, and actually those numbers would then mean something in terms of therapy. So it wasn't cool that something that was not really backed up with tissue would be used for any type of decisions. And I got messages from colleagues that the management doesn't like people who cause conflict, that this is not collaborative. And then other pathologists would join the group and the group would grow, and like people would not go to me because they didn't think like I was really telling them the truth, what's in the tissue, and they would go to another pathologist, and oddly enough, they would tell them the same thing because they are trained pathologists and they see what's in the tissue. So that didn't work that well. But yeah, I became the problem pathologist. So that was there at that place, and it ended up well. We then developed SOPs. We had a special team. It became a robust workflow, and we worked with the statisticians for tests to have a robust validation. And then I moved on to a different company, to Charles River Laboratories. That was around that time where I met Tom at Pathology Visions, and I looked at their platform. It was a nice platform, and then it turned out that the Charles River group had collaboration with Aiforia, and I got to work in Aiforia, and I was so happy to see this validation feature there. I'm like, wow, you like have a setup to do this. That's fantastic. But what I have noticed that there was already some work going on, but that this because this is still I don't know if we can call it cutting edge still. I guess it's still cutting edge, digital pathology in general. It was still cutting edge. So I saw this at Charles River and at different places that people were developing this workflow that we did at my previous company. So we decided to publish a paper on this, and here's the paper as proof. We published Developing Qualification and Verification Strategy for Digital Tissue Image Analysis in Toxicological Pathology. We published, and Tom is on this paper. I'm the first author, and colleagues from across the industry and academia. We put together our know-hows and our experiences, hurdles that we have to go through from what happens to the tissue, from the problems that you can have with the tissue, with the staining, with the team. And something that I want to focus right now is the team. So Aiforia is giving the possibility for people to just use it out of the box, or I love that it's cloud-based. This was super cool. I come from an environment where for like every time there was an upgrade, all the computers had to be upgraded. And I mean, it's not image analysis software specific, but it's super handy that this is cloud-based, right? But the team, at some point, now, I don't know how many of you do the annotations for your project on your own. Yeah, so most of you. So you have the know-how, you know what you're annotating, right? And you know also how very scalable that is. Not too scalable, right? So I was the one annotating everything at the beginning, and then we had this QC team, and for repetitive tasks and for things where people started getting the knowledge what's in the tissue, the pathology group started training other people to do annotations as well. So there was a training element involved to teach the person who is training this algorithm or developing this algorithm what the structures of interest are, how they should be annotated, what they mean, what the endpoints are of the algorithm, and basically giving a person who doesn't have the same background and the same versatility of tissue understanding, let's put it that way, teaching them the important thing to be able to work on this particular project. Then in terms of annotations, that would be one group, and then there would be a group that would work with software. Here, in this case, you can be like one-man band, you can do everything there. But if you do more projects, often you end up in a supervisory role, and then other people are doing things for you. So one of the things that you have to do, or that has to be done, is teaching the people working with you on the project that they are doing the right thing. So you have 3 validators always, right? How very much do they agree at the end? And those validators, by the way, are experts. They are already experts. Let's say they would be pathologists or neurologists or somebody who has the same background as you, right? You trust them, they will understand correctly. How much difference do you see still when they are annotating? There is still difference, right? Gillian said like 70, 80, we agree. That's a pretty good agreement. This is the level of agreement that you can achieve, or the difference there is maybe a little bit of a difference in interpretation. There is another type of difference that you can have that kind of compounds is when the definition of ground truth, the definition of your annotations is not clear enough to everyone. you can end up with a lot more difference than you need by everybody annotating things correctly, but not really in the way that is good for the validation. So we developed a retinal atrophy model, where in the retina, you have a nuclear layer in the retina that is the outer nuclear layer has a lot of nuclei, right? And we would do object counting for this. So first we had to agree, okay, when do we count those nuclei when they overlap? When do we count them as separate? Okay, when they touch, yeah, separate. When they do like this, are these separate? Do you only count one? And like there was a whole discussion because even though everybody knows how this nucleus looks like, it's easy, then you end up with differences from like this interpretation. At the end, we said like 50% overlap, you still count two. If it's more, you just count one, you can pick whichever. Because we would end up with like 50% difference in counts because of this thing, right? Everybody was an expert, was well trained. So training in the understanding of tissue, training in the understanding of how we generate ground truth and how it should be later reproduced for validation and verification. And then this is going to be for generating your numbers to check image analysis model against your annotation. So you can do quantification. What we also ended up seeing that this only covers a part of your image, like you put your fields of view or the regions where you want to count. I mean, you're not gonna not anymore regularly, but often got a request, oh, can't you annotate a full slide? Like, no, I cannot. I cannot annotate a full slide. Unless it's, a week label for the slide, positive or negative for marker. But like, no, I cannot annotate all the cells in the slide. So for the quantitative validation or verification or whatever word we use, basically checking if the algorithm, the markups match the tissue, you only have parts of the image, right? What's feasible in a reasonable time by an expert that's gonna be contributing to this. But there's another part that we incorporated in our process is we call it visual QC, where you look at the whole image, not really annotating particular things, but checking if the signal that you are quantifying, if the markups match relevant things in the tissue. And then if it does, all good, proceed to the drawing and to the validation. If not, then we have to go back to the drawing board and improve them all. But basically, this process was something that we described in this paper. This can be applied, and this is super cool because in Aiforia, that's basically incorporated in the workflow, how you do it. But that's something that can also be done in a GLP, good laboratory practice environment, where you may be using other tools in addition. If you want to do stereology in addition to do 3D data or whatever, you can use this process of training people, making sure that everybody is on the same page regarding the tissue structures, making sure everybody is on the same page regarding what structures and how we are annotating, doing the quantitative QC, visual QC, and by an independent expert that was not involved in the development of the algorithm. Why did I have this conflicting situation? Why did it get so emotional? Because I was working with people who were very much vested into the development of the algorithms. And we are biased to love very much what we are doing and think it's fantastic. And then, no matter how professional you want to be, we are psychologically wired to defend our creation. So that's why it's important that the final validation verification is done by somebody who did not produce this algorithm. So that's that. And even though it's published in ToxPath, it applies to everything image analysis. So I highly recommend reading this paper. Thank you very much.
You stay till the end. It means you are a real digital pathology trailblazers. All the links are going to be in the notes below, but there's something that I want to give you right now. It's the Digital Pathology 101 book. We just recently published this book. It's available for free, and I want to give it to you so that you can be successful in this digital pathology and tissue image analysis space faster. It has everything you need to know to start and continue your digital pathology journey, including tissue image analysis. So don't wait, grab it from the link below. And if you know somebody who would be interested, please share it with them. And I talk to you in the next episode.